In this video, we're going to talk about the pancreas. The pancreas is a gland that's found just posterior to the stomach, and it has both exocrine and endocrine functions. The exocrine functions of the pancreas are mediated by these acinar cells, which produce these enzyme-rich digestive juices that we'll talk about later on in the digestive system. Now, the exocrine function of the pancreas comes from little islands of cells called pancreatic islets, also called the islets of Langerhans. These contain endocrine cells that release different hormones, like alpha cells and beta cells. Now, the alpha cells of the pancreas secrete the hormone glucagon, and the beta cells of the pancreas secrete the hormone insulin. Now, both glucagon and insulin are antagonistic, meaning that they oppose the effects of each other. What we see here is a microscopy image of a pancreatic islet, and it's this sort of lighter stained tissue here. And so this whole lighter stained tissue right here is a pancreatic islet or islet of Langerhans, and the cells within the pancreatic islet are going to be alpha cells and beta cells. Now, you don't need to be able to recognize the difference between alpha cells and beta cells, but what's more important to know is that alpha cells in the pancreatic islet release glucagon, and beta cells release insulin. Now, in this particular microscopy image, we can also see nearby the pancreatic acinar cells, which secrete enzyme-rich digestive juices. So the first hormone we'll talk about here is glucagon. Now remember, glucagon is going to be released by alpha cells of the pancreas, and the major target of glucagon is on the liver. Now, ultimately, the effect of glucagon is that it causes an increase in blood glucose levels. And it does this through two different mechanisms. Glucagon can stimulate a process known as glycogenolysis, as well as another process called gluconeogenesis. Now, glycogenolysis means the breakdown of glycogen into glucose. So glycogen, if you remember from chapter 2, is a polysaccharide, or basically a long chain of glucose molecules which allow your liver to store glucose for later use. Now, lysis means to break down, so glycogenolysis means the breakdown of glycogen. And so glucagon will actually stimulate the breakdown of glycogen into glucose and then help liberate some of that glucose into your bloodstream to ultimately raise blood glucose levels. Now, what's also interesting is that glucagon can also stimulate something called gluconeogenesis. Gluco means sugar, neo means new, genesis means to make. So gluconeogenesis means to make new sugar or make new glucose. And so what happens here then is that glucagon actually stimulates the synthesis of glucose from lactic acid and other non-carbohydrates. Now, through this process of gluconeogenesis, glucagon is actually able to raise blood glucose levels by making new glucose and then incorporating that into your bloodstream. So the effects of insulin are actually opposite to that of glucagon. And so what we'll see then is that insulin is a hormone that lowers blood glucose levels, whereas glucagon actually increases blood glucose levels. Insulin will lower blood glucose levels. Now, the way that insulin lowers blood glucose levels is that it enhances the membrane transport of glucose into fat cells and muscle cells around your body. And insulin also helps inhibit glycogenolysis and gluconeogenesis. Now, what's interesting is there are other effects of insulin, such as participating in neuronal development and being involved in learning and memory. So not only does insulin help lower blood glucose levels, but it also plays a role in learning and memory. Now, not all cells in the body require insulin to have glucose transported into their cells. In fact, liver cells, kidney cells, and brain cells don't require insulin to have glucose uptake, whereas the rest of the cells in your body require the hormone insulin in order to get glucose into their cells. Now, the way that insulin exerts an effect on target cells throughout your body is that it binds to a special type of receptor called a tyrosine kinase receptor. Now, this tyrosine kinase receptor is also an enzyme, And once you activate this enzyme receptor in the plasma membrane of target cells, this leads to an intracellular signaling cascade that ultimately causes increased glucose uptake. So basically, the transport of glucose from the extracellular fluid inside of the cell through the plasma membrane, that's going to be stimulated by activation of these tyrosine kinase receptors. Now, some of the other effects of tyrosine kinase receptors is that it's going to trigger other enzymes to begin oxidation of glucose to make new ATP. It will also stimulate the polymerization of glucose into glycogen. So taking glucose and putting it into long chains to form the molecule glycogen. And that glycogen is a way you can store glucose for later use. It can also convert glucose to fatty acids, and this is going to primarily occur in adipose tissue. Now, what we see here on this slide is basically just showing a negative feedback mechanism for blood glucose homeostasis. So what we see here is that we're going to have normal blood glucose levels, which are about 90 milligrams per 100 milliliters. Now, you don't need to memorize this particular value, but it's just important to note that their blood glucose levels are a homeostatic variable, so that we need to maintain pretty normal blood glucose levels. The reason being is that cells throughout your body, require glucose to, function and make new ATP, and especially your brain. So if your blood glucose levels drop too low, then your brain cells don't have enough glucose to function properly. And so what you see then is if your blood glucose levels drop too low, is you can enter into diabetic coma, which is basically a loss of consciousness due to low blood glucose levels. Now, what we see here on this slide is basically just going to be the mechanisms which act to maintain homeostasis or balance of blood glucose in your body. So let's first start with the imbalance of decreased blood glucose levels. So let's say if you undergo hypoglycemia or low blood sugar, low blood glucose levels, what's going to happen is that alpha cells in the pancreas will be able to monitor the blood concentration of glucose in your body so that if your blood glucose levels get too low, those alpha cells will release the hormone glucagon. Now remember, glucagon is a hormone which acts to increase blood glucose levels, and it does this by primarily acting on the liver and initiating the breakdown of glycogen into glucose, or glycogenolysis, as well as gluconeogenesis. However, that's not on this slide. Now, glucagon having effect on the liver, which causes glycogen to break down into glucose, that's going to allow glucose to be released from the liver back into your bloodstream and then increase your blood glucose levels back to a normal homeostatic range. So the important point to note here is that glucagon increases blood sugar levels by stimulating the liver to release glucose reserves to raise your blood glucose level back up to homeostasis. Now the opposite of this is going to be if you have increased blood glucose levels, such as hyperglycemia or blood glucose levels beyond the normal homeostatic range. And this can happen. This can occur during, like after eating a meal, where your blood glucose levels will spike. Now, what's going to happen here then is that beta cells in the pancreas will monitor blood glucose levels as well. And so, if your blood glucose levels get too high, such as in instances of hyperglycemia, those beta cells will release the hormone insulin. Now, insulin is a hormone which acts to decrease blood sugar levels, and it does this through a variety of different mechanisms. The two main mechanisms that are illustrated on this particular slide is that insulin is going to stimulate the uptake of glucose by cells throughout your body, so that if you can actually stimulate cells to allow glucose to go into the cell, you're effectively removing glucose from the bloodstream. Now, insulin also stimulates the conversion of glucose into glycogen within the liver. So ultimately, your liver can help remove glucose from your bloodstream and store it in the form of glycogen for later use. And this is in effect mediated by the hormone insulin. Now, both the uptake of insulin in cells as well as the conversion of glucose into glycogen in the liver both act to decrease blood glucose levels, which is going to help allow you to decrease your blood glucose levels back to a normal homeostatic range, as you can see here. And so from this particular slide, you can see that there are antagonistic effects of glucagon and insulin. In fact, they both act to help maintain glucose homeostasis in the body. Now, you may have all heard of a disease called diabetes, which is fairly common. Now, there are some cardinal signs of diabetes mellitus, and some of the cardinal signs of diabetes mellitus include polyuria, polydipsia, and polyphagia. Polyuria means high urine output, lots of urine. And the reason why individuals with diabetes have polyuria is that glucose acts as an osmotic diuretic, meaning that if you have high glucose levels in your blood, ultimately what happens is that glucose may be found in high concentration in your urine. Well, because glucose itself is a solute, that's going to cause water to follow glucose into urine because water follows solutes, and therefore you get high urine output because you get more water loss due to the movement of glucose through your kidneys. And this is going to cause excessive urination or polyuria. So one of the symptoms of diabetes mellitus is this polyuria, which causes you to urinate a lot. In fact, individuals will wake up throughout the night having to urinate, and so they won't get a good night's sleep as a result of this symptom. Now, polydipsia relates to polyuria, and this is going to be the excessive thirst that comes from increased water loss. So if you're urinating a lot, that means you'll be losing a lot of water, which means you're going to also be thirsty. So polydipsia is going to be excessive thirst in response to diabetes mellitus. And so you're also going to just drink a lot of water or drink a lot. Now, one of the other symptoms of diabetes mellitus is polyphagia, or excessive hunger and food consumption. Now, the reason why polyphagia comes about is that cells throughout your body, if they cannot get glucose into the cell, then effectively your cells will be starving. Whether or not your blood glucose levels are high or low, if you can't get that glucose from the bloodstream into your body's cells, your cells are going to be effectively starving. Now, they can release signals that then stimulate your brain to feel hungry, so that if your cells are starving of glucose, they'll actually cause your brain to undergo excessive hunger. So then you'll eat a lot just to try to get more glucose into your body cells, even if you have high blood glucose levels.